Lecture Notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 58. 16. Inheritance. Genetic information is transferred from parents to offspring to maintain the continuity of life. In sexual reproduction, meiosis introduces genetic variation so that offspring resemble their parents but are not identical to them. Genetic crosses reveal how some features are inherited. The phenotype of organism is determined partly by that they have inherited and partly by the fact of the environment. Genes determine how organisms develop. Gene control in bacteria gives us a glimpse of this process in action. 16.1 Passage of information from parents to offspring. The production of gametes for sexual reproduction occurs through meiosis. The 4 resultant daughter nuclei are haploid, so that the fusion of gametes at fertilization restores the diploid number. Anaphase 2 is the separation of the daughter chromatids by pulling apart centromeres, producing 4 haploid cells. Meiosis increases genetic variation through chiasmata and the independence of chromosomes during metaphase I. The law of segregation states that pairs of factors determine the characteristics of organisms, but only one factor of the pair enters a gamete. These factors are variations of a gene, called alleles. The law of independent assortment was Mendel's second law. This states that either member of a pair of alleles can combine with either of a second pair during gamete formation. Gametes contain one sex chromosome, so makes a heterogametic, producing X and Y sperm, whereas females are homogametic. Human blood group is controlled by multiple alleles. IA and IB show codominance over each other with IA recessive to both. Genetic is the study of inheritance, i.e. how characteristics are passed from one generation to another, via reproduction. The production of gametes for sexual reproduction occurs through meiosis. This is the division of a nucleus following chromosome replication, to produce daughter nuclei, involving 2 successive divisions. The 4 resultant daughter nuclei are haploid, contain half as many chromosomes as the diploid parental cells. Metaphase I begins when the nuclear envelope breaks down and the bivalence line up on microtubules, called spindle fibers, at regions called kinetochores along the equator of the cell. The second meiotic division occurs in both cells produced during division I. In prophase I, chromosomes condense and become visible again. The chromosomes then become attached to the spindle fibers and line up individually at the equator of the cells in metaphase II. Anaphase II is the separation of the daughter chromatids by pulling of the centromeres. Finally, telophase I produces 4 genetically different, haploid cells, following cytokinesis. Meiosis results in genetic variation within a population. Lecture Notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 59. The independence of chromosome arrangement on the spindle at metaphase I results in the number of possible chromosome combinations in a gamete being very large. Chiasmata linkage results in crossing of genetic material between chromosomes. Chiasmata are random and produce genetic recombinants, new combinations of genes. 16.1 to 1 haploid, N, and diploid, 2N. During meiosis, cells with 2 copies of each chromosome, diploid cells, divide to form cells with only 1 copy of each chromosome, haploid cells. Diploid cells contain sets of chromosomes and therefore 2 copies of each gene. In sexual reproduction, haploid gametes are formed containing 1 set of chromosomes and therefore 1 copy of each gene. Each offspring receives 2 copies of each gene, 1 from each of its parents. 16.1 to 2 homologous pairs of chromosomes. During prophase I of meiosis, homologous chromosomes pair up to form bivalents. These are linked at points along their length called chiasmata. Characteristics are controlled by pairs of alleles, which are found at similar loci on homologous chromosomes. Lecture Notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 60. Alleles for different characteristics behave independently when carried on separate pairs of homologous chromosomes. This leads to independent assortment of characteristics. In humans, normal cells contain 46 chromosomes, 23 homologous pairs. Gametes contain 23 unpaired chromosomes, so that the fusion of gametes at fertilization restores the diploid number. This conserves the number of chromosomes, since diploid gametes would double the number of chromosomes every generation. The first stage of meiosis is prophase I. Chromosomes, visible as two daughter chromatids, condense and homologous pairs come together, forming bivalents. These bivalents are linked at points along their length, chiasmata. The homologous chromosomes separate during anaphase I, with one member of each pair moving to the poles. Telophase I is the final stage of the first division and is the reverse of prophase I. Cytokinesis, cytoplasmic division, occurs, with filaments of the proteins actin and myosin contracting to produce cleavage furrow at the equation of the cell. This produces two cells containing one member of each homologous pairing. Each chromosome is still made up of two daughter chromatids. 16.1 to 3 Need for reduction division. Meiosis consists of two divisions. The first division, meiosis I, is a reduction division that separates the homologous chromosomes, so that each cell now has only one of each pair. The second division, meiosis II, separates the chromatids of each chromosome. Meiotic division therefore produces 4 cells, each with one complete set of chromosomes. 16.1 to 4 Behavior of chromosomes and nuclear envelope during meiosis. The typical behavior of chromosomes in meiosis is that homologous pairs synapse, recombine, and then separate at anaphase I. At anaphase II, sister chromatids separate. However, studies of small chromosomes in maize derived from a variety of sources typically have failure of sister chromatid cohesion at anaphase I. The mechanism of nuclear envelope breakdown, NEBD, was investigated in live cells. Early spindle microtubules caused folds and invaginations in the NE up to 1 hour prior to NEBD, creating mechanical tension in the nuclear lamina. In addition to NEBD and reformation, the nuclear envelope has a specific function in prophase I of meiosis, the anchorage of telomers. In animal, yeast, and some plants a chromosome bouquet is formed and anchored to the nuclear envelope via the telomers. Lecture notes for A-Level Biology by Mohammad Rashid Chatha. 030046100457. 61. 16.1 to 5 Photomicrographs of meiosis at different stages. 16.1 to 6 The crossing over and random orientation, independent assortment. The cells produced by meiosis are genetically different from each other and from their parent cell. This results from independent assortment of the chromosomes as the bivalents line up on the equator during metaphase I, and also from crossing over between the chromatids of homologous chromosomes during prophase I. Genetic variation also results from random fertilization, as gametes containing different varieties of genes fuse together to form a zygote. A gene found on the X chromosome but not on the Y chromosome is known as a sex-linked gene. Genes that are close together on a chromosome that is not a sex chromosome are said to be autosomally linked. 16.1 to 7 Random fusion of gametes. Meiosis, random mating, and the random fusion of gametes produce genetic variation within populations of sexually reproducing organisms. Variation is also caused by the interaction of the environment with genetic factors, but such environmentally induced variation is not passed on to an organism's offspring. The only source of new alleles is mutation. Lecture notes for A-Level Biology by Mohammad Rashid Chatha. 030046100457. 62. 16.2 The roles of genes in determining the phenotype. Mendelian genetics are patterns of inheritance worked out by Gregor Mendel crossbreeding garden peas in the 19th century. The simplest crosses are between two individuals which differ in one characteristic, monohybrid crosses. Mendel made the following deductions from this cross: parental characteristics are not blended, as there are no medium-sized plants produced. This led to Mendel's first law, the law of segregation. The characteristics of organisms are determined by pairs of a factor, with only one factor of the pair entering a gamete. These factors are known as alleles, defined as alternative forms of a gene, i.e. the gene for height can be tall or dwarf. Let T equals gene for all, dominant, and T equals gene for dwarf, recessive. Because homologous chromosomes are separated at anaphase 1 of meiosis, during gametogenesis pairs of alleles are separated. The consequence of this is gametes contain only one member of each pair of alleles. Fertilization combining gametes results in new pairs of alleles forming. A backcross or a test cross is used to determine the genotype, alleles present, in individuals with a dominant phenotype, physical appearance, i.e. a tall individual could be genotype TT or TT. The dominant phenotype individual is crossed with a recessive individual, whose genotype must be TT. If the F1 generations are all tall, dominant parent's genotype is TT, homozygous, ratio 1:1 tall, dwarf, dominant parent's genotype is TT, heterozygous. Dihybrid crosses study the inheritance of two pairs of characteristics. In the cross below, the parents are homozygous for both characteristics. Mendel made the following deductions from this cross: since the F1 are all tall and red, these characteristics are dominant over dwarf and white respectively. In the F2 generation, two new combinations appear, tall and white, and dwarf and red. Therefore, the alleles for height and color can be separated. The F2 ratio for tall, dwarf and red, white are both 3:1. Therefore, the alleles are behaving independently. This led to Mendel's second law of independent assortment. Either member of a pair of alleles can combine with either member of a second pair in gametogenesis. In the dihybrid cross shown, either of the alleles for height can be in a gamete with either for flower color. Therefore, the F1 generation has 4 possible gametes. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 63. 16.2:1 gene, locus, allele, dominant, recessive, codominant, linkage, test cross, F1, F2, phenotype, genotype, homozygous and heterozygous. A gene is a length of DNA that codes for the synthesis of a particular polypeptide or protein. Only about 10% of a chromosome consists of genes, the rest is non-coding DNA. Diploid individuals contain two alleles controlling most characteristics, with one allele inherited from each parent. These pairs of alleles are found at the same position, or locus, on homologous chromosomes. Independent assortment, alleles for different characteristics behaving independently, occurs when they are carried on separate pairs of homologous chromosomes. The homologous pairs line up independently on the equator during metaphase 1 of meiosis. It is a matter of chance that which combination of alleles move to a pole together and go into a gamete. Human blood group is controlled by multiple alleles. There are 3 alternative allele forms, IA and IB show incomplete dominance over each other, codominance, with IO recessive to both. Phenotype. Blood group, ABOAB. Genotype IAIA or IAIOIBIB or IBIOIOIOIAIB. The genotype of an organism showing dominant characteristics can be determined by looking at the offspring produced when it is crossed with an organism showing. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 64. Recessive characteristics. This is called a test cross. Monohybrid crosses consider the inheritance of one gene. Dihybrid crosses consider the inheritance of 2 different genes. Different genes may interact to affect the same phenotypic character, a situation known as epistasis. Mendel identified recessive characteristics that one parent may show, which do not appear in the F1 generation, but then reappear in the F2 generation. He concluded that individuals contain 2 factors for a characteristic. Dwarf characteristic is recessive and tall is dominant, because no dwarf plants occurred in the F1 generation. The reappearance of the dwarf plant in F2 means that the F1 plants, which are tall, must contain 1 factor for tallness and 1 for dwarfness. Mendel observed that when two individuals differing in one single characteristic were crossed, the first generation, F1, all resemble one of the parents. If any F1 individual is then self-pollinated, the second generation, F2, may have either of the original parent's characteristics, in an approximately 3:1 ratio of F2 generation. A backcross is used to determine the genotype of individuals showing the dominant phenotype. This individual is crossed with a recessive individual. An organism's genetic constitution is its genotype. Its observable characteristics are its phenotype, which is influenced by the expression of its genes. Different varieties of a gene are called alleles. Alleles may show dominance, codominance, or recessiveness. An organism possessing two identical alleles of a gene is homozygous. An organism possessing two different alleles of a gene is heterozygous. If a gene has several different alleles, such as the gene for human blood groups, these are known as multiple alleles. 16.2-2 Genetic diagrams. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 65. 16.2-3 Interpretation and Construction of Genetic Diagrams. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 66. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 67. 16.2-4 Test cross. A Punnett square is used to show possible offspring from the random fertilization between these possible gametes. A backcross or a test cross is used to determine the genotype of an individual with dominant characteristics. For example, a tall, red plant can be TTR, 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 or TTR. The sex of an individual is determined by a pair of sex chromosomes X and Y. The X chromosome is much larger than the Y, and since the Y is much smaller, it carries very few genes. In mammals, including humans, females are 20, homozygous, and males are XY, heterozygous. However, in some organisms e.g. butterflies, males are 20 and females are XY. In meiosis, sex chromosomes pair up and are separated to opposite poles of the cell. Therefore, since gametes only contain one sex chromosome, males are heterogametic, producing X and Y sperm, and females homogametic, producing X ova. A chromosome is a large, densely staining body consisting of a single, supercoiled DNA strand. The genes along a chromosome are found at particular points called loci. The genetic code is the sequence of bases contained in the DNA molecule. The code is read in groups of 3 bases, called codons. Each codon codes for a particular amino acid. The central dogma, principle, of molecular biology is that genes are read by messenger RNA, which in turn moves out into the cytoplasm where it codes for the assembly of a protein. At the ribosome, messenger RNA attracts transfer RNA molecules with the right amino acid attached. The amino acids are held together so that peptide bonds form. In this way the protein is assembled. DNA replication occurs when the 2 strands separate and the enzyme DNA polymerase assembles 2 new strands on the originals. This is called semi-conservative replication, as each old strand is conserved as half of the new DNA. A fault in DNA replication leads to a gene mutation. The base sequence is altered so that the gene no longer codes for the correct protein. Chromosome mutations. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 68. Can result in whole blocks of genes being lost, or translated backwards, for example. 16.2-5 chi-squared, chi-2, test. The chi-squared, chi-2, test can be used to find out whether any differences between expected results and observed results of a genetic cross are due to chance or whether the difference is significant. The separation of homologous chromosomes takes place during the first meiotic division. During the second meiotic division, sister chromatid of each chromosome are separated into daughter cells. Because of the way that the members of different pairs of homologous chromosomes separate independently of each other during the first meiotic division, independent assortment of homologous chromosomes, meiosis produces daughter cells that are genetically different from each other. In general, if there are n pairs of homologous chromosomes, independent assortment will produce 2n different combinations of chromosomes in the daughter cells. This genetic variation is increased because chromatids break at points where they intertwine, chiasma, and join to the other homologue. Monohybrid crosses involve He inheritance of a character controlled by a single gene. Dihybrid crosses involve the inheritance of characters controlled by two genes that occur at two different loci. 16.2 to 6 Relationship between genes, proteins, and phenotype. The HBB gene codes for the beta-globin polypeptides in hemoglobin. An allele of this gene with a different base sequence produces sickle cell hemoglobin. The allele of the HTT gene that is responsible for Huntington's disease includes a repeated triplet of nucleotides called a stutter. Albinism and hemophilia show the effect on the phenotype of missing or inactive polypeptides. Specifically, tyrosinase is a key enzyme, which catalyzes a rate-limiting step of the melanin synthesis, and the downregulation of tyrosinase is the most prominent approach for the development of melanogenesis inhibitors. 16.2 to 7 Role of gibberellin in stem elongation. Gibberellins promote growth in different ways. These hormones are mainly involved in controlling and promoting stem elongation, flowering, and leaf expansion as well as seed germination. They are used in suspension cultures to enhance the growth of cells. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 69. 16.3 Gene control. Any diploid cell has two copies of the gene controlling a single character. If both versions of the gene, called alleles, the same, his individual has a homozygous genotype. If the two alleles of the gene are different, the individual has a heterozygous genotype. In presenting genetic crosses, we use a standard format that shows phenotype of parents, genotype of parents, gametes, genotype of offspring, 1, generation, expected frequency of phenotype of offspring, 1, generation. If the parents are purebreeding, homozygous, the offspring, 1, generation is sometimes called the F1 generation and the offspring, 2, generation is sometimes called the F2 generation. An allele of a gene is said to be recessive if it fails to exert its effect in a heterozygote. The allele of the same gene that exerts its effect in the heterozygote is said to be dominant. The alleles are codominant if they both show their effect in a heterozygote. Sex is determined by sex chromosomes. A character unrelated to sex is sex-linked. If it is controlled by a gene whose locus is on the female sex chromosome. The chi-squared test, chi2 test, helps us to determine whether observed results fit the expectations we had based on a null hypothesis. As a general rule, we accept any variation from what we expected so long as its probability is greater than 0.05. The evolution of warfarin resistance in population of rats. Sequence of events leading to natural selection. Application of these events to the evolution of warfarin resistance in populations of brown rats. 1. There is competition within the population for environmental resources that are limited. Members of a rat population compete for food. 2. There is genetic variation within the population. A random gene mutation caused one rat to become resistant to the pesticide, warfarin. 3. One genetic variant confers an advantage on the possessors of a favorable allele of gene. The rat with the allele for warfarin resistance would not suffer hemorrhages where warfarin was used, whereas a rat with only alleles for warfarin susceptibility would suffer hemorrhages. 4. The possessor of the favorable allele will have more offspring than the possessor of only the less favorable. Being stronger and healthier than its susceptible competitors, the resistant rat. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 70. Alleles will successfully raise more offspring. 5. The possessor of the favorable allele will pass this allele on to some of its offspring. As a result, the frequency of this allele in the population will increase, i.e. natural selection has occurred. The resistant rat passed on the resistance allele to some of its offspring. As a result, there were resistant rats in the offspring generation. Repeated over several generations, the warfarin resistance allele became very common in the rat population. When taking samples, errors can occur through chance or through human bias. We can reduce these errors by taking more than one sample and by using random sampling techniques. Data collected by sampling from a population can be represented as a frequency distribution. A normal frequency distribution is a special type of distribution in which the mean, median, and mode have the same value. The standard deviation is a measure of the way that data are spread out from the mean. The more the data are spread out from the mean, the greater the standard deviation. The standard error, SE, is calculated from the standard deviation of a sample and is used to make allowances for the difference between samples and the total population from which they were taken. 95% of data lie within a range plus 1.96 SE of the mean. An organism's phenotype results from an interaction between its genotype and factors in its environment. Whereas some characteristics are influenced by a single gene, others are influenced by many genes. This is called polygenic inheritance and gives rise to continuous variation. Variation exists between the members of any species. This is caused by process that occurs during meiosis and by gene mutation. The causes and effects of the three sources of genetic variation. Source of genetic variation: how it causes variation effect. Crossing over during prophase I of meiosis changes the combination of alleles of one or more genes that are carried on a chromosome, and therefore the overall combination of alleles on that chromosome. Genetic variation between gametes produced by an individual. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 71. Independent assortment during metaphase I of meiosis. Random orientation of bivalence results in many different combinations of chromosomes and therefore many different combinations of alleles. Genetic variation between gametes produced by an individual. Random fusion of gametes: any male gametes can fuse with any female gamete. Genetic variation between resulting individual organisms. Hormone site of production: some notable effects. Auxin embryo, young leaves and growing points, particularly apical bud. Promotes cell elongation, apical dominance, C assignment, inhibits formation of lateral buds. Gibberellins embryo, young leaves and growing points breaks seed dormancy, promotes germination and seed development by stimulating cell elongation. Cytokinins roots promote cell division. Abscisic acid root cap, older leaves, stem imposes seed dormancy and inhibits germination. Ethylene ripening fruit, older tissues promotes ripening of fruit. Plants fall into one of three categories according to their photoperiodic response. Short day plants need long nights. They won't flower until RFR leaves reach a threshold. These plants generally flower in the summer-autumn, as the nights get progressively longer. Long day plants need short nights. They won't flower unless the period of darkness is lesser than a certain critical length each day. Day-neutral plants use some other environmental or internal factor to trigger flowering. 16.3 to 1 difference between structural genes and regulatory genes. The lac operon provides an example of how a prokaryote can alter the transcription of a cluster of structural genes coding for enzymes concerned with lactose uptake. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 72. And metabolism, depending on whether or not lactose is present. Structural genes code for the proteins required by a cell for its structure or metabolism, whereas regulatory genes control the expression of other genes. A repressor protein can block the synthesis of a repressible enzyme by binding to the gene's operator site. An inducible enzyme is synthesized only when its substrate is present. Regulatory gene: a gene that regulates the expression of one or more structural genes by controlling the production of a protein, as a genetic repressor, which regulates their rate of transcription. A regulator gene, regulator, or regulatory gene is a gene involved in controlling the expression of one or more other genes. Regulatory sequences, which encode regulatory genes, are often at the 5' end to the start site of transcription of the gene they regulate. 16.3-2 Genetic control of protein production in a prokaryote. The primary method to control what type of protein and how much of each protein is expressed in a prokaryotic cell is the regulation of DNA transcription. All of the subsequent steps occur automatically. 16.3-3 Transcription factors. Prokaryotes use operons to control the expression of genes in cells. Eukaryotes also use transcription factors to control gene expression. A transcription factor is a protein that controls the transcription of genes by binding to a specific region of DNA. They ensure that genes are being expressed in the correct cells, at the correct time and to the right level. 16.3-4 The way gibberellin activate genes. Transcription factors in eukaryotes make sure that genes are expressed in the correct cell, at the correct time and to the correct extent. In plants, gibberellins allow gene transcription by causing the breakdown of DELA proteins which inhibit the binding of transcription factors. Keywords: albinism, allele, anaphase I, anaphase II, autosomal linkage, chi-squared, X2, test, codominance, crossing over, degrees of freedom, X2 test, deletion mutation, DELA protein repressors, dihybrid inheritance, diploid, 2N, dominant allele, embryo sac, plants, epistasis, F1, F2, fertilization, embryo sac, plants, gametes, gametogenesis, gene expression, gene mutation, genetic cross, genetic variation, genotype, gibberellins, hemophilia, haploid, N. Heterozygous, homologous chromosomes, homozygous, Huntington's disease, inducible enzyme, insertion mutation, lack operon, low allele, locus, meiosis, metaphase I, metaphase II, monohybrid cross, phenotype, pollen grain, prophase I, prophase II, Punnett square, recessive allele, regulatory gene, repressible enzyme, sex linkage, sexual reproduction, sickle cell. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 0300461047. 73. Anemia, structural gene, substitution mutation, telophase I, telophase II, test cross, transcription factors, tyrosinase. Chapter Glossary. Albinism is an autosomal recessive condition due to mutations of the tyrosinase, TYR, gene. It is characterized by lack of pigment in the skin, eyes, and hair. Allele is a version of a gene. Anaphase I is the third stage of meiosis where the chromosomes that make up the bivalent are pulled apart to the poles of the cell by the spindle fibers. Anaphase II is the seventh stage of meiosis where the chromatids are pulled apart to opposite poles of the cell. Autosomal linkage is the condition when two or more genes are positioned on the same autosome. They are unlikely to be separated by crossing over during meiosis, so are often inherited together. Chi-squared, X2, test is a statistical test used to determine whether a pattern of inheritance is statistically significant. Codominance is when both alleles for a gene in a heterozygous organism equally contribute to the phenotype. Crossing over is the exchange of genetic material between two chromosomes in a bivalent. Degrees of freedom, X2 test, is the number of categories -1. Deletion mutation is a form of gene mutation in which one or more nucleotide bases are removed from a DNA sequence. This may lead to a frameshift mutation, changing every successive codon. DELA protein repressors are negative regulators of gibberellin. Dihybrid inheritance is the determination of a trait by the inheritance of two genes. Diploid, 2N, are cells with two copies of each chromosome. Dominant allele describes an allele that is always expressed. Dominant alleles are represented by a capital letter in genetic crosses. Embryo sac, plants, is the female gametophyte in flowering plants. It is formed when the haploid megaspore nucleus divides. Epistasis describes a relationship between genes at different loci, where the allele of one gene affects the expression of a different gene. F1 is the first generation of offspring from a genetic cross. F2 is the offspring produced when F1 individuals reproduce with one another. Fertilization: the fusion of male and female gametes. This is random and produces genetically different individuals. Gametes are sex cells that have a haploid nucleus and are produced through meiosis. Gametogenesis is the process of producing new gametes that occurs in sex organs. Gene is a length of DNA on a chromosome that codes for the production of one or more polypeptide chains and functional RNA. Gene expression are some genes in cells are switched on so that their corresponding protein is produced, whereas other genes are silenced. Gene mutation is a change to at least one nucleotide base in DNA or the arrangement of bases. Gene mutations can occur spontaneously during DNA replication and may be beneficial, damaging, or neutral. Genetic cross are individuals of a species are bred. The ratio of phenotypes present in their offspring can be linked to the genotype of the parent. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 74. Genetic variation are differences in the DNA sequence of individuals, including individuals of the same species. Genotype is an organism's genetic composition. Describes all alleles. Gibberellins are plant hormones that control stem elongation, trigger the growth of the pollen tube during fertilization, and stimulate the mobilization of food storage reserves during seed germination. Hemophilia is a sex-linked genetic condition caused by a mutation of the factor VIII, F8, gene on the X chromosome. This disrupts the body's ability to form clots. Haploid, N, are cells with only one copy of each chromosome. Heterozygous is when someone has two different alleles of a gene, e.g. FF. Homologous chromosomes are two chromosomes with similar gene loci but different alleles, one inherited from each parent. Homozygous is when someone has two identical alleles of a gene, e.g. FF. Huntington's disease is a genetic condition caused by a mutation, CAG trisomy expansion, of the Huntingtin, HTT, gene. This results in progressive death of nerve cells in the brain. Independent assortment. Independent assortment is a source of variation in meiosis where the bivalent chromosomes can line up either way around on the metaphase plate. Inducible enzyme is an enzyme which is only expressed when cells are exposed to its specific substrate. Insertion mutation is a form of gene mutation in which one or more nucleotide bases are added to a DNA sequence. This may lead to a frameshift mutation, changing every successive codon. Lac-operin is a group of three structural genes, LAC-Z, LAC, and LAC-A, that are required for the metabolism of lactose. LA allele is a dominant allele that codes for a functional enzyme in the gibberellin synthesis pathway. If a plant possesses the LA allele, gibberellin is active and the plant grows tall. LA allele is a recessive allele that codes for a non-functional enzyme in the gibberellin synthesis pathway. If LA is homologously present, the plant remains short. Locus a position on a chromosome. Meiosis is a type of cell division used to produce gametes that produces four genetically different haploid daughter cells from one parent cell. Metaphase I, the second stage of meiosis where the bivalent chromosomes align along the metaphase plate and independent assortment occurs. Metaphase II, the sixth stage of meiosis where the recombinant chromosomes align on the metaphase plate. Monohybrid cross is a genetic cross in which only one gene is studied. Phenotype is an organism's observable characteristics. It is due to interactions of the genotype and the environment. Pollen grain is a pollen microspore that the male gametophyte arises from. Prophase I is the first stage of meiosis where the nuclear envelope breaks down, the spindle fibers form and the chromosomes condense and form bivalents. This is the stage of meiosis where crossing over occurs. Prophase 2, the fifth stage of meiosis, where the nuclear envelope breaks down, the spindle fibers form and the chromosomes condense. Punnett square is a square diagram used to predict the possible genotypes and phenotypes of an offspring and the probability of each when two individuals breed. Lecture notes for A Level Biology by Mohammed Rashid Chatha. 0300461047. 75. Recessive allele describes an allele that is only expressed in the absence of a dominant